Okay, folks, shall we get started? Welcome to everybody in the auditorium and those online. It's a pleasure for me to introduce you Associate Professor Jessica Mar from AIBN. Many of you know Jessica, but for those who are not so familiar, I'll tell you a little bit about her background. So Jessica did her Bachelor of Science in Mathematics at the University of Queensland, and then did her honors here. And then she went to do a PhD at Harvard in biostatistics, so Harvard University in Cambridge, Massachusetts. And she obviously liked Cambridge so much, she stayed there for a little bit longer at the Dana-Farber Cancer Institute. And then she moved to the Albert Einstein College of Medicine in New York, where she was for nearly 7 years. And during that time, she started a joint appointment at the AIBN from 2016, and she's been full-time with UQ since 2018. And so many of you will know Jessica from her work, but also from collaborations. I know the people are very much engaged with what you're doing, Jessica. So I won't give a summary of your work, but I'll let you do that. So we very much look forward to your talk. Thank you.
Thanks a lot. Thanks so much, Paul, for the really kind introduction and also the opportunity to come visit. I'm embarrassed to say it's taken 2 years to make it over to TRI to give a talk. So it's wonderful to finally be here. Also, I looked at my calendar and this is the first in-person talk that I've given all year. So it's such a privilege and a pleasure to be here. I think it's also probably going to be the only one this year. So I'm really excited to be here for many reasons. And a special shout out to everyone who's online. We can see your chat, but I look forward to getting questions as well from you later on in the talk. So I'll just jump straight into it. The title of my talk is right here. And basically what I'm going to be talking about is heterogeneity. So, oops, oh, this one. Okay, we're in business, great. So I think an enduring theme in my career that I found really, really fascinating that I keep coming back to is this idea that cells are inherently heterogeneous. And I think we're all under no illusions now that cells are different from each other, but certainly growing up through the years, different kind of ensemble methods that we've had have really painted this picture that cells are uniform. And so it's really exciting to see, for instance, the uptake of these single cell sequencing technologies that have really revamped how we understand transcriptional regulation and more broadly, all kinds of streams of biology. So in my group specifically, as Paul mentioned, my background is in biostatistics. And so what I find really fascinating is using the distribution to really inform our understanding of what's going on inside cells. So if you imagine that this is a, oh, sorry, I was going to use my mouse so everyone can see. Okay, so if we look at this density here, I've used this example for a single cell, a population of single cells, but really any point along this density can represent a patient or a sample that you're interested in. And so in statistics, we have these estimators called moments, and they tell us about the structure of the data. So I'm sure you're all familiar with the first moment, which is the mean or the median. It's the central location here. But we can look at other moments. So the second moment, for instance, is the variance or the spread of this distribution. And we can continue on to higher moments. So for instance, the third moment is the skewness. We can go up to kurtosis, etc. And as they go up higher, these moments lose their interpretability. But the point is that they report on interesting things because we all know that the most interesting biology happens at the extremes. And I think what's a really cool source of innovation is to consider that most of the methods that we have in bioinformatics typically focus on what's going on at that first moment. And so during my career, we've really looked at different aspects of this and how they can tell us interesting things about biology. Certainly, as we think about the shape of this distribution, we also can't ignore different features like bimodality, multimodality, and different things like that can also really tell us about extreme values, outliers, and different subclusterings that are going on in the data. So I think this is a really cool area to study. And way back when I was a postdoc, I wanted to try out this idea as a proof of principle. And really, we were looking at variance, so the second moment, the spread of the distribution, as a way to understand regulatory parameters in the context of human disease. So if you look at two groups here. So we've got a disease group and a control group. And what we often do when we have gene expression data is we rank those genes in terms of how different they are on average. And this statistic up here is a t-statistic. And so if you're familiar with this, you'll know that the numerator here is the difference between the two averages, right? So genes that are more differentially expressed would have a larger difference here on average. And their variability, which is on this numerator, would be, the smaller that is, the larger the t-statistic, the more significant this result is. And this is a really insightful way to rank or prioritize gene expression. But the issue is that it ignores some of these other scenarios that could also be important. So if you look below this gray line, there are these two cartoons that I've outlined. So for instance, one situation where there is a difference in average expression, but there is much larger variability for this gene profile in the disease group compared to the control. And the more extreme example over here, where there is actually no difference in average expression, but the variability is quite different between these two. So we tried this idea out in the context of stem cells, olfactory neural stem cells taken from Parkinson's disease patients and schizophrenia patients. That was working with Professor Alan Mackay-Sim at Griffith, and we published a study a while ago. So I won't go into it except to highlight that there are some really interesting things that can come from looking at statistics that go beyond that first moment. So I'll start by talking about one of the studies that we got involved in a while ago, and this was looking at human embryonic development. So this is data that was published in this publication here in 2013 by this group in China. And what was really cool is they looked at single-cell RNA sequencing for developing embryos. And while there wasn't a lot of cells, as you can see from these numbers, what I thought was really cool is that we had every single cell in the human organism. So for our data, we took this from 4-cell embryo up to blastocyst. And we wanted to ask a few different questions. But before I get into that, I want to highlight that working with public data can also be a huge minefield. And I'm sure if people have had that experience before, they can relate to some of the challenges. And so before we jumped into the kind of questions that we wanted to ask from the science, there were a lot of different checks and balances we had to do to ensure that the data was appropriate for our needs. So I'm just highlighting, these are the sorts of things that we did. I'm happy to go into it if anyone's interested. But the kind of things we wanted to ask of this particular study was, what can variability really tell us about transcriptional control during human embryonic development? And so I had a really talented student who just finished undergraduate come work with me at Albert Einstein College of Medicine. And we asked this question around different directions of variability. So if you think about genes that are very stable, they have very invariant expression from cell to cell. Excuse me, I keep losing my mouse. From cell to cell within an embryo. What does that tell us? These are genes that are highly homogeneously expressed. They could be really predictable for telling us about the cell state when we sample them from the embryo. Concurrently, if we think about genes that are highly variable, these are the genes that are very heterogeneous from cell to cell from the same embryo. And this might reflect the diversity of different states for the cells that become different cell fates. And so we constructed some really simple statistics to basically pick up these two extremes of variability. And we found a few different things, but two things I want to focus on, or one thing I want to focus on, is we use this modeling to identify new stage-specific markers. And so if you look at the data or the embryo as it's transitioning from 8-cell stage morula to blastocyst, the usual way of how we find biomarkers is to look at changes in average expression. And this might be well and good, but there are likely to be more subtle changes going on when you consider every single cell in this growing embryo. So what we wanted to do was overlay an additional requirement based on variability. So we asked that a potential marker be elevated on average for a specific stage, but it also had to have the least variability for that stage compared to others. And when we put that proof of principle into practice, we found different kinds of markers for each of these stages. And you can see by the box plots, this is how they were distributed overall. But it's really much more informative to look at this on an individual gene basis. So these are the single cell gene expression profiles for the variability markers that we found using these new rules for the blastocyst stage. So for each marker, each dot represents a single cell and the expression in that cell at the different stage. The blue dots represent the average, and you can see how it's changing and really delineating that blastocyst stage. So among these 11, I think, that we found, we're excited to see EpCAM. EpCAM is a potential marker for blastocysts. It's also known to be involved in the formation of trifectoderm. So that was exciting to see that come up. We took this to our collaborator, Ernst Wolvertang, at AABN, and he wanted to choose the gene that we knew the least about to validate. And he and his team validated this gene HDDC2 in human iPSCs and ESCs. So what they did was they knocked it down using shRNA. And what they saw in those knockdowns was the repression of NANOG and DMT3B. These are pluripotency factors in these iPSCs. They then overexpressed HEC2 using CRISPR-Cas9. And what they saw was an increase then in NANOG and PAX6, which is a differentiation marker. So it seems like this gene was involved in maintaining the pluripotency in these cell lines. Obviously, if we could validate further, we could say more. But I think this was really exciting to see this kind of result come out of a computational prediction based on variability in this phenotype. So we know that, well, this was focused on a single gene, but we know that at the same time, cells are regulated through gene networks. And we know that no gene is operating in isolation. So part of our job as bioinformaticians is to really figure out what these regulatory networks are and how they're really important in transitioning cells from one stage to another. So some of the other work we've done has been to really look at the ability to reconstruct these networks reliably. And a few years ago, what we saw was the application of these existing network methods to single cell gene expression data. And it was almost a bit of a concern. We weren't sure if really this was the right thing to do. So I had a really talented PhD student, Shunan Chen, who wanted to set up this kind of benchmarking study where we took different network reconstruction methods that were general, but also there were some single cell specific methods too. And we took some real data as well as some simulated data to really see if these network methods were doing a good job in reconstructing gene networks for single cell data. And I'll just cut to the punchline of whether they were any good. And based on our evaluation, it seems that the evidence is really not supporting this as a valuable thing to do. Of course, it's never that straightforward. I think when we test out different bioinformatics software, the methods did have different strengths and weaknesses, but not in any direction that we could really point to. Obviously, there's a lot more network reconstruction methods that are coming out every single day, which I think is what makes working with single cell data somewhat challenging. But I think this really drives home the aspect of horses for courses and being aware of the strengths and limitations of your tools. So we've looked also at variability in the context, not just networks, but also pathways. And a tool that we worked on a while ago, this is my postdoc in New York, Dr. Laurence Detrante, and our programmer, Sam. And really the idea here was to capitalize on the distribution of variability that can occur within a pathway. So if you imagine we have a patient population and this very standard signal transduction pathway, there is a distribution there in the variability, and information on genes that are much more lowly variable versus those that are much more highly variable could really indicate how that pathway is under different degrees of regulatory control. So there's some very simple statistics that underlie how to detect these sorts of changes. This was published a few years ago, and essentially we can test two different kinds of comparisons. So the idea of looking at a pathway, its distribution from low to high levels of variability, and comparing that either to a control or a reference group, we can ask whether this overall profile is any different. And we can also ask whether specific levels of lowly variable genes, for instance, is different from a reference or control. So I won't go into that anymore, but to show you an example of how this works, we've applied this in the context of breast cancer, where we've got data from the Cancer Genome Atlas. And here we're capitalizing on the matched design that is inherent in TCGA. So we've looked at match control and tumor samples. And this is just one of the pathways that came up as significant. It shows us what we would expect to see, I think, where we see more diversity or more heterogeneity in the tumor samples relative to the control. But it's a good way to illustrate how this package is working. We've just been invited to participate in a COVID study from New York City, looking at patients that they've done RNA-seq on, and they'd like to try to see if this kind of method can identify inflammatory inflammation type signatures. So it's exciting to try to apply this tool in different situations to learn more. So one thing I will do is explain also just how one aspect of bimodality, where we've looked at bimodal gene expression, can really help us understand breast cancer in a different light. So as you might know, continuous data can really be represented by different kinds of statistical distributions. So we're all familiar here with the normal distribution, right? But there are some other distributions out there too. So for instance, the Cauchy is very normal-like with heavier tails. We also have log normal, which is a bit more skewed, the Pareto, which is very skewed, the gamma distribution, and also the bimodal distribution. And so a question that I thought was really fascinating was to ask whether there really is non-normality in cancer transcriptomic data. So we often make this assumption that gene expression either, well, I guess less so now, follows a normal distribution, but follows a distribution. And we rarely stop to question whether different genes might follow different statistical distributions themselves. So we wanted to test this out, again, using the Cancer Genome Atlas data. We selected 3 different studies, so AML, glioblastoma, and ovarian cancer. And the goal of using these 3 different tumor types was really to get at the large sample size that is in TCGA. So these are nearly 200, 500 patients big. What my team did was we developed this pipeline, which looks a little bit complicated, but really what it's seeking to do is to put or to classify, each gene into one of these distributions. And based on our statistical tests, if there wasn't a good fit, then the gene wasn't included in this analysis. We didn't try to force any gene to belong to one of those 6 distributions. And so how did the results shake out? So this was data where we actually had microarray data as well as RNA-seq data. And what's really interesting is we could look at the platform effect as well on these different tumor types. So you can see for microarray, the different colors represent the different distributions that these genes followed. And I think the most interesting thing to note is that the normal component is certainly not overwhelming. And you can see that there is a bit of a platform effect, but what's really interesting too is that these are the same patients that are in both of these 2 groups. But certainly as we move to RNA-seq, while the normal component grows, it's certainly the non-normal components don't necessarily go away. So this is a really interesting study. There were other things that we found, but what I wanted to use this for is to really highlight, the fact that there were bimodal expressed genes in these different tumor types. So I had a really outstanding MD PhD student, Dr. Daniel PK, who's now doing his residency at UCSF. And he really wanted to follow up this idea of why there was bimodal gene expression, and could that tell us anything about the breast cancer cohort that we were looking at. So what we mean by bimodal is this kind of two-peak shape, where it really reflects in this context a group of patients that have expression of the gene at this lower level versus another patient group that has higher expression for the same gene. So this is a real example that we pulled out from the Cancer Genome Atlas. And the context that we were interested in exploring this idea was, could we use this idea to find new oncogenes? So this idea that if we looked at matched tumor normal data and we found genes that were bimodal in the tumor and unimodal in the normal, but to the point where the expression was much lower, this could be an interesting candidate for a theoretical oncogene, right? Obviously for only those patients that were in this group. So what we did was set up the data to look for this. We had a few different statistics to try to tease out this profile, based first on selectivity, to try to find that separation between the overexpressing tumors and the normal group. We also then ranked these genes based on how overlapping and cleanly separated they were. And as you can see, these are the 5 profiles for the top 5 allele genes that fit these specific statistics. So what's really interesting about some of these genes is that they have, 2 of them have prior roles in oncogenesis, so CBX2 and NEL2. We also showed how if you use more traditional tools, we don't find these. It's really looking at that bimodality relationship that we were able to find these 5 genes. We wanted to understand and see if they could help explain what was different between these 2 cohorts. And so we developed a lasso-based model to basically integrate lots of different features. Those kinds of features were these things on the right-hand panel here. So basically the idea was to use this particular gene to separate out the 2 groups, but then to look if there was anything predictive about these sorts of features that defined whether a patient was more likely to be in this peak versus this peak. And so there's a lot to unpack from this diagram, but essentially for each of these biomarkers, we can see that this predictive model worked in some cases for the AUC really well, like for CBX2, and in other cases for this gene EPYC, we had a fairly bad predictive performance. So I think that reflects the nature of these genes and what they do. But to focus on CBX2, what we saw was the patients that ended up in either mode were really predictive in terms of promoter methylation, in terms of intronic methylation, and also the particular subtyping of the tumor. So just to show you here a bit more what that means, this is the intronic DNA methylation, and you can see for patients in this group, this is overall the beta values of the methylation values that this group had versus the group that were in the lower mode. So this shows you the separation that we see. What this has to do with CBX2 is really interesting, but this is also how we're sort of breaking down the relationships from this data. If you go back into TCGA and look at overall survival, you can actually see there's a significant difference between patients that were in that lower mode versus the upper mode. And that is significant based on the log-rank test for the survival, so that's interesting. The other thing we wanted to ask too is, was there any correlation with the aggressiveness of the molecular subtype? So when you break the patient groups down into their subtyping, so luminal A, B, HER2, and basal, and you count the number of patients that were in the high versus the low group, we see that it does actually track with aggressiveness of the subtype. And what's really interesting too is this assignment of these subtypes is based on this signature, this AIM signature that's been computationally derived. The gene that we're focusing on, CBX2, is not actually part of this algorithm. So it highlights potential utility then for using this gene to potentially identify subtyping, especially of these aggressive tumors. The other thing we wanted to know was, in normal tissues, how does expression of CBX2 play out? And so using GTEx, we were able to scan across a lot of different adult human tissues and healthy donors. And you can see that CBX2 is really only highly expressed in adult testes. So as a marker for breast cancer, this is actually not a bad thing. So the final thing we did was to partner with colleagues at Albert Einstein College of Medicine, who then took human MCF7 breast cancer cells and knocked down CBX2. And we saw that there was significant slowing of growth of these cells, which was quite exciting to see. Again, as this came from a computational prediction. So this method is, although we've piloted on cancer data, it's certainly not restricted to looking at that. But through this, we've found a few different interesting things. And if you're interested in checking it out, this is available in Bioconductor and was also published last year in BJC. So the last area I will talk about is that I think one of the really interesting things about coming to Australia was to be able to focus more on a new area of research for my group, which was to look more into understanding how humans age. And in the last 2 years that I've been at AIBN, what's been really fun is to set up some new projects where we ask this question in a few different ways. So we are working on some mouse studies where we've used metformin, which I'll talk about a little bit more today. But metformin is a drug that's been shown to extend lifespan in model organisms. And so we're really interested in understanding the transcriptomic profile behind that. We're also interested in understanding how telomere attrition disrupts the transcriptome of aging cells. So we know that as we get older, telomeres get shorter, but thinking about single cells, how does that happen? How is that distributed across the cell population? And how does that impact the noise or the heterogeneity that we see in the transcriptome? We've also started looking at images, which is a really brand new direction for my group. But we're hoping to develop machine learning methods based on these images that we're capturing to try to make predictions about whether cells are older or younger, whether cells are senescent or not. We all know that biological age is not the same thing as chronological age. So trying to get a handle on whether we can extract more information from images to make those predictions is a direction that we're taking. And of course, we know that as we get older, there are a confluence of other diseases that come along. And understanding the aging effects of that, looking at cancer, osteoporosis, and Alzheimer's is an area that we're just getting started in as well. So let me just briefly describe some of the experiments that we've done recently, which are quite exciting. So this was done by my PhD student who's now graduated, Dr. Meir Kolkani. He's working at AbbVie in Chicago now. But we were working with Professor Nir Barsalai, who's director of the Institute for Aging Research at Albert Einstein. And through some generous sources of funding, we were able to do quite a large single cell sequencing data set. And we took mice, 4 mice from each group. So we had young mice, we had old mice, and we had old mice that were given metformin. So what's so fascinating about metformin is that this is an FDA-approved medication. It's usually used for type 2 diabetes, but in model systems, they've shown an extension of lifespan, although the in vivo mechanism is not completely clear. We also don't know how different tissues respond to metformin, especially in the context of aging-related pathways. So what we did was we took adipose and muscle. These are 2 tissues that are quite relevant for aging. And we sequenced them using single cell sequencing assays. And let me tell you a little bit about the data, even though we haven't completely understood what's going on yet. So in adipose, what we did was we broke this down into major cell types. So if you're familiar with single cell RNA sequencing data, you'll know that there are many steps coming from the data that comes out of the machine into being able to interpret what's going on. And so if anyone's interested in that, I'm happy to speak to those steps that we found useful in the group. But we've used a method called single R, which leverages existing data sets to classify the profiles that we see into the cell types that we know. And just to highlight, so this was adipose, but this was stromal vascular fraction that we took from these mice. So the adipocytes were too large, I think, to go through the 10x sequencer, But nevertheless, you do see these adipose-derived stem cells, macrophages, immune-related cell types that are really interesting. What we also were trying to look at was the effect of metformin. And I know it's hard to make out, but that's also the point where you see that really the treatment effect, which is here in green, is not glaringly obvious. We do see an overlay here with the B cell group, which is quite interesting, but it's not like we can point to massive clusters that are appearing just because of the treatment effect. And perhaps we expected to see that. Obviously, an aging phenotype is not super dramatic, especially for the transcriptome. And so that just lends itself to more analysis to figure out what's going on. We've also done this for muscle, but I didn't include this because it's a fairly similar profile and just to keep the story straight. So we're trying to figure out what's going on through much more standard analyses in terms of genes that are specific for these different cell types, genes that are specific for the old and the treatment conditions that we've applied. But one thing I wanted to highlight too is to go back to this idea of different distributions. So the story I told you earlier was in the context of bulk level data, but we're also really interested in single cell gene expression data too. And I think in this case, what's really interesting is that in single cells, we have lots of zeros to deal with. As anyone knows, they can be quite challenging. And so the nature of using different kinds of distributions to model these gene expression profiles, I think the challenge is even greater. And what we tend to do again with single cell RNA-seq data is assume the presence of a single distribution. So whether it's a Poisson distribution like this or a negative binomial distribution, or we often use these methods that allow for the zero inflation that we tend to see. But again, we don't stop to look to see if specific genes might follow one distribution over another. So I have a really great student, PhD student, Melindri, who's taken on this challenge to understand the prevalence of these different distributions in single cell data. And so here, what we do is we assume that the profile is gene specific, and we try to determine the most appropriate fit for each gene expression profile based on either a combination of these different choices. And so Melindri comes from a stats background, and what she's developed so far is a framework to essentially test for which distribution, zero inflated like this, or Poisson or negative binomial, is the most appropriate for the genes that we're modeling. And essentially the idea is to then be able to classify which is the most appropriate fit. So just to show you briefly, what we've got here is the first question of whether the standard assumption that we make, which is the negative binomial distribution, how well does it explain all the genes that we're looking at? And so one of the advantages in the group is to be able to generate these really novel data sets like the metformin one that I explained, but also to be able to apply some of these really novel kind of bioinformatic approaches to ask new questions about the data. So for adipose, what she's found is that although the majority of genes are actually appropriately fit by negative binomial, there is certainly diversity in some of the other candidates. So these are what these percentages reflect. And just to show you, this is the old group from the mice that I showed you before, the metformin treated group and the young group. So you can see that as we scan across these different conditions, the percentages do change, but they're not predominantly dictated by one condition versus another. For muscle, strangely enough, we see much fewer negative binomial genes, and then the increase in some of these other distributions. So this is really interesting because what it also lends the next questions of is whether the differences in distributions are also telling in genes that are involved in regulating these conditions. So for instance, if a gene were to change from negative binomial in old to Poisson in the treated, that's really interesting. And even more so, the change from these unimodal genes to the zero inflated ones, that tells us that there are many more cells in this new condition likely where we have dropout or less transcription of these particular genes. So comparing treated versus old also tells us about metformin's drug action and whether we actually get this reversion back to a younger phenotype. So this is, I guess I finished my talk earlier than I thought, but this is the final slide so far that we have. I just got this a few days ago, so this is really new results that we're still trying to make sense of. And this is the idea that when we look at genes that are statistically significant in changing their distribution, not necessarily their average expression, but their distribution from one condition versus another, where are those, which cell types are affected by those specific changes, especially with the conditions that we're comparing. So I know this is a bit of a busy slide, but if you just focus here, this is the data for adipose. And what we've done is we've calculated how many differentially distributed genes there are changing between old versus young. And what you see down here is basically the pairwise comparison between the different cell types. And the red indicates that this comparison is significant. It means that we got many more genes than we would expect by chance. And if you look, the comparison here is between T cells and B cells. So when you look about these differentially distributed genes in T cells and B cells, the overlap is much higher than we expect by chance. What's really interesting is then when we look across to the similar comparison between old and metformin mice, we see again the significance in these overlapping differentially distributed genes that are occurring in these T cells and B cells. Looking at muscle, we see the same thing, except these are regulatory T cells, and that could reflect the classification system that we've used for this data set as well. So what does this all mean? Obviously, there's a long road to go in figuring out what these differentially distributed genes are. But I think this slide really highlights how looking outside the box also opens up these new questions. I think when we look at single cell data, there are far more subtle effects going on. And so we really need to ask these questions about changes and shifts or changes in distribution rather than focusing only on average expression and how those changes might be occurring. So I will summarize my talk. I know I've talked about a lot of different aspects, but that's really to bring you along and sort of explain how throughout my career, I've been really interested in understanding how heterogeneity occurs in gene expression and also what it can tell us about understanding transcriptional regulation. So looking at embryonic stem cells, using this to identify new markers of different stages that we otherwise might have missed. I think the two cancer examples also illustrate how, again, modeling distributions is really important. It helps us identify those extremes, but also in the CBX2 example, how clustering of these substructures can identify the relevance of specific oncogenes that are relevant for those groups rather than looking across the whole population as a group. And finally, can modeling heterogeneity in single cell data sets provide insight for the aging process? I think that is still a question, but I think it is a really exciting work in progress. We do have some other studies in the group where we looked at single cell sequencing for mesenchymal stem cells that we've been following. We have some other models that I think are interesting there, not just looking at mice. But I think overall modeling heterogeneity in aging is a really fascinating question that really gets, I think, at the more basic mechanisms of deregulation as humans age. So I will end there in the hope that we have lots of time for discussion and questions. But I do want to acknowledge the two groups that I ran, who I've mentioned in the course of this talk. So I had a really great team at Einstein. They've all gone on to their amazing careers in their next chapters. But certainly Shunan Chen, Daniel PK, and Laurence de Charente were really absolute pioneers in pushing work forward. We've also had the privilege of working with some really great clinical collaborators over the years. And at AIBN and at UQ, it's really no different. We've built up a great critical mass, I think, of people in terms of my students and obviously my postdoc, Dr. Ati Tahirian Fad. And again, we've really leveraged, I think, some of our local collaborators. But as with anything, we are seeking new collaborators as well. So it's been a really great day so far at TRI to meet new people, and looking forward to more engagements. So thank you very much. I'm surprised that the talk went so quickly, but hopefully there's lots of questions on Zoom and here with our audience too. So thank you. Do you want to do the flip? I'm not quite sure how to do that. Oh, fantastic.
Online. So we have a question from Ankit. Ankit, would you like to ask your question? Or if you prefer, I can read it for you. Ankit, so we had a question from you. Would you like to ask your question, or if you prefer, I can. Oh, let's see. Okay, so it says, could you comment on how well to do the normalization methods? This is very technical. Yeah, I don't even know what I'm saying here. i.e., TMM used by EdgeR, RLE used by, how do you say that? DSEC2 or TPM work for single cell differential gene expression data.
That's a good point. Okay, so thanks for the great question, Ankit. It sounds like you've done some research into these different methods already. In our group, we've been using Seurat to do differential gene expression. And I wish my team was online now. They could tell you exactly the kind of normalization. But I'm not clear if that's done internally. It also, I think, there are a few different ways to do that differential gene expression within Seurat. But I think the methods you're describing, EdgeR and DSEC2, I mean, they certainly work for RNA-seq. So in terms of, I think they are also being used for single-cell RNA-seq. But I can't really comment on the effectiveness of those normalization methods. So, but I think, yeah, I would encourage you to look at Seurat if you haven't already done so. So yeah, sure. So thanks, Matt. It's a shame you're not here. So Matt asked, has anyone looked comprehensively at epigenetic heterogeneity? Yes. So I believe your group has been looking at that too.
It's obviously a very important question.
It is a very important question. We have not worked. Okay, yeah, no, no, it is a really important question. I think also for DNA methylation, you know, it makes a lot more sense for different distributions, different states to be in play because of the way, you know, regions are methylated or not methylated or hemimethylated, rather than looking at gene expression. So there are people that have looked at heterogeneity. I'm blanking a little bit, but I know Rafa Irizarry, he's done a lot of looking at heterogeneity and DNA methylation. So that's really interesting. I brought up your work not to be facetious, but also because I think, you know, looking at different distributions for DNA methylation for studying heterogeneity and TCGA data was a really interesting question that I know one of Matt's students has been asking. So I believe there is, but I think there's obviously more things to be doing, and also linking it to gene expression. I know that's a little bit challenging. We did try to do that to some degree, but I don't think we were 100% successful at it. Certainly with the assays that we have now, and some of the coupled assays too, looking at methylation and RNA-seq in tandem, I think it would be really interesting to look at the nature between epigenetics and transcriptional heterogeneity together.
We have a question in the room, so Mitch.
Yeah.
Thanks, Jess. I had a question about CBX. Is that correlated from the
I don't believe so. Yeah, so for everyone at home, Mitch asked a really great question about CBX2 and whether it correlated with any of the clinical parameters. So I'm just trying to bring up this. So that was something we did look at to a degree. So we did look at some of the clinical parameters here, like with stage and like copy number variation. But you were just asking before about, what was the other part of your? That's right, yeah. No, I don't believe so, except we did look at these subtypes. So when you break the patients down into their subtypes, so HER2 positive, for instance, and you count the number of patients that were high for CBX2, so they're in the upper mode, there was a different proportions of these patients as you move through the subtypes, and that correlated with the aggressiveness of these subtypes as they're known to be. But we didn't dig into that relationship anymore. So yeah. No, that's true.
Thank you. Nicholas, do you have a question? Same question.
It's a good question.
So do we have any more online? No. Can I ask you a question about metformin?
Yes.
So I suppose what I didn't really understand is how you think heterogeneity would be important in the response there.
Yeah.
So the data on metformin and aging,
Mm-hmm.
it's a bit controversial, especially whether it would be true in humans, I think.
Correct.
But there's also this information, that metformin might also suppress cancer.
Yes.
Because of the effects that it has through AMP kinase. So, but what I suppose what I'm not really understanding is how heterogeneity might play a role in that. How you see that might be important.
Yeah, that's true. So, oh, right, sure, sorry. So, it's a good question. I think with any drug, especially for something like metformin, where you take it over a long time, I think heterogeneity in the sense of cellular uptake, like is every cell in that tissue being affected by metformin? What is that distribution? Are different cell types similar in their uptake profile, in terms of how the transcriptome is changing? I think that's the angle that I was interested in. Obviously, that's much more generic. It's not specific to metformin itself. But I think with aging, as people age, there are different patterns in terms of tissue specificity and how that degradation, if you like, occurs. So I was curious to know how, overlaying a drug like metformin, which is known to have some effects, like what that would look like. So, yes.
So, I mean, you pose this very interesting idea that you might get more heterogeneity in gene expression in cancer. Mm-hmm. But do you think that would be a normal part of aging, is that you start to see a bigger differential in gene expression between individual cells?
Yeah, that is a really great question. I mean, I think, so it's interesting because I think earlier on I would ask people who work in aging, what do they think? Like as people age, do you expect more variability from cell to cell? Or do you expect the other way, to have less variability? And it's really interesting because people were split. So people were very, very adamant that, no, you get increased heterogeneity because you get reduced effectiveness of signaling pathways, things start to go wrong, they accumulate, cellular senescence kind of throws the spanner in the loop. And other people will say, no, you get more homogeneity and there's more fragility or an ability to tolerate noise. And so I think really it's a combination of both. I think it depends on what genes or what pathways you're looking at. And I think that tissue specificity of it is really interesting too, because we know the kind of symptoms that crop up in aging are sometimes quite isolated to specific organs, specific tissues. And so I think it is really interesting to understand the distribution of that variability. I don't think it's that straightforward.
We have a question from Antje.
Oh, yeah. Same mice, yes.
Might be best if you repeat
Yeah.
it rather than me.
So Antje's question was, you know, in the last study where we looked at, and I guess this is only for the audience, but in terms of looking at adipose and muscle from the same mice, yet we see really different profiles. What is behind that? And oh, thanks. So I think that speaks to, first of all, and I think I'm glad you asked that because now I can actually show you, although, thank you, not side by side, but so this was the breakdown of the different profiles in adipose, but I think I have this at the end where, right, so this is for muscle. And yeah, you do see a real, like this, there's representation of the similar cell types, but there are other ones as well, and there's a real flattening, I think, of the variation. Certainly the kind of treatment and the aging effects we see are not exactly the same. I think this really speaks to the fact that transcriptomes are cell type specific, tissue specific. And I think when you drill down at the single cell level, you're picking up on all those variations. So we do actually have bulk data from these mice, not the same mice, but similar strains of mice who've gone through these treatments and the same tissues. So I think it would be interesting to look at. What we've, yeah, no, it's not quite the same thing, but in looking at here, I know it's impossible to see, but in comparing the different cell types, what we've also tried to see is when you look across like all of the data and not break it down into cell types, whether we also see those distributions popping up. And for the large part, we don't. So I think that also again speaks to the cell type specificity of these transcripts really doing different things. So in some ways that's fascinating. I don't know how you find that though, like whether that's a confronting assumption or mm-hmm. Yeah. Mm-hmm. Yeah. Yeah. Well, I think it's really interesting to hear you say that because I think that's also how something like this kind of model where I think it's so important, right? Because say flipping from this to this, you know, in the presence or absence of, you know, a drug or a treatment or, you know, an inflammatory response, that then tells you a lot about this gene's role. But if we make an assumption that everything sort of follows this with variations, it's not to say you can't pick up on that, but I think intentionally sort of studying that flip could uncover some really interesting biology. So, because then also it allows us to drill down and say, well, which cells are in this component versus this component? You know, what are the cells doing on this side? They may have different states because this is just a single gene. Are there other things that could indicate, you know, altered behavior, altered pathways that are then really informative? So. Any more questions, I think?
We've got any more online? I think we're done if there's no more questions online. So just remains to thank Jessica for a really interesting, informative talk. Thanks very much, everybody, for joining us online. And Jessica will still be around this afternoon for, she's meeting with a number of people, and so you may well meet her coming around the building. Okay. Thank you very much. Thanks, Jessica.
Thank you. Thanks, everyone. And I'm always on email as well if people want to follow up with more questions. So thanks so much today.